Howdy Pima Aztecs, welcome to Module 2B, Chapter 3, Cells, Part 4. So there's 5 parts in Chapter 3, Cells. So there's a lot of information about cells. So here's, you guys know this cartoon if you've been reading or paying attention to the videos that I've made for the cells. We're going to talk about cell replication today. So this is when you have a single cell and you want to replicate, aka duplicate it, and make 2 daughter cells. So what's all the cool molecular biology that goes on with cell replication? Here's the diagram that we've been studying. Parts that we'll need for cell replication, aka duplication. So we'll zero in on these guys, the centrioles, and we'll need some microtubules. We'll need the DNA to make a copy of itself so that each daughter cell can have 1 full set of chromosomes. So, basic cell physiology. Cells have the ability to do a lot of things. They can, if you are a cell, you can metabolize things. So that has to do with, for example, taking in glucose, chopping it up, and making ATP. If you are a cell, you can digest food. Even if you're a single cell, you ingest food and then you digest it, or you break it down into usable parts. You dispose of waste. Again, even if you're a single cell, you have to dispose of the waste products. And we're going to focus on this video on reproduction of cells. Also, cells have the ability to grow, move, and respond to a stimulus. This is a cartoon of the cell cycle. This is from Benjamin Cummings. And in the olden days, we used to talk about the cell cycle like the dial on a washing machine, but washing machines don't really have dials anymore, but we can still sort of look at it that way. So you know how you have a life cycle? So you're born, you're a kid, you grow into a teenager and an adult, and then you die. So you have a human cycle, and all the cells in your body have a cell cycle. So, and this is a cartoon of it. These parts on the outside would be a cartoon of a single cell. And then this would be on the outside here, this would be a cartoon of 2 daughter cells about to split. So they haven't quite split yet, but you've got the chromosomes here that have been duplicated, and you're about to split into 2 daughter cells. And in this cartoon outside of the cycle, it shows the chromosomes lining up and getting ready to split into the 2 daughter cells. So for example, this could be daughter cell 1 and this could be daughter cell 2. So if you are a cell and you're just living your life, you're going through a cell cycle, you have this outer part. This whole outer green arrow is called interphase. And interphase has actually 3 subphases or 3 parts. Interphase has G1 phase, that's where you're growing. The S phase, S stands for synthesis, because you are still growing as a cell, but you're also duplicating or replicating your DNA. And then another word for that is you're synthesizing a second set of DNA so that each daughter cell gets a set of DNA. And then the other subphase within interphase is called G2. You're still growing. You can see in all these interphases you're growing your cell. And then in G2 you're making the final preparations for cell division, aka mitosis, or the mitotic phase. And then you can see this little sliver here. These are the stages. You guys probably remember this from high school or junior high. I think I learned it in kindergarten because I was at a fast-paced kindergarten. Just kidding. I wasn't. I didn't learn this in kindergarten. I'm kidding. High school, I think. So mitosis is divided into these phases: prophase, metaphase, anaphase, and telophase. And then this could be a tricky test question. This piece here, cytokinesis. So kinesis is movement, cyto is cell. Cytokinesis is when the cells actually split or they move into two separate daughter cells. So cytokinesis technically is not part of mitosis. But that's where it gets a little weird, but this whole mitosis and cytokinesis are all part of the mitotic phase. But if you're asked a test question, name the subphases of mitosis, you would say prophase, metaphase, anaphase, telophase. Okay, so the cell cycle, like I said, includes the changes a cell goes through from the time it is formed until the time it reproduces, just like you, the life cycle. From the time you are formed, when the sperm meets the egg, until the time, well, this is where it's a little bit different, where until the time you reproduce, you know, that's where the cell cycle and the human cycle are different. Because some people don't ever reproduce, but they have a full human cycle and they live to be 100 years old. So the cell cycle includes interphase, and then included within interphase are G1, S, and G2. 90% of a cell's lifetime is spent in interphase, so about 10% is spent in mitosis. But, however, there's always an exception to every rule. Some cells are stuck in GO. And on this cartoon, it doesn't show GO here, but there's a little piece in here called GO, and that means you are stuck there. They never divide. Can you think of some cells that would apply to? Yeah, if you said your nervous system cells, yeah, because when you destroy your neurons, for example, and become a paraplegic or a quadriplegic, those cells can't regenerate. They can't form new daughter cells. Mitotic cell division, this is another way to look at it. Mitotic cell division is the same thing as mitosis, results in each daughter, the daughters are over here, having exactly as many chromosomes as the parent. So it doesn't show, but this would be a parent cell here, and it would have all the chromosomes in there. And after you duplicated them, each daughter cell would have, for example, in this cartoon, 1, 2, 3, 4. 1, 2, 3, 4. That doesn't show up very well in the cartoon. But anyway, each daughter cell should have 4 chromosomes, because that's how many the parent had. Just a couple more details on chromosomes. So here's a cell, here's the nucleus with the chromosomes in it, and then they enlarge a chromosome here. And you can see chromosomes have banding patterns on them. And at the end, they have these pieces called telomeres. And you can see if you unwind a chromosome, it's just made up of DNA. The chromosome is made up of DNA and proteins. But in this cartoon, it's just showing the DNA unwound. So telomeres are found at the end of chromosomes. They're implicated in aging and cancer, so a lot of scientists are studying how telomeres shorten over time to understand cancer. Okay, here's our cartoon of the cell cycle. We've kind of mentioned these, but cell G1, you're a cell, you're in G1. You're just doing your job, being a cell. You're duplicating your organelles and adding more cytoplasm. So you're duplicating your organelles because eventually you'd like to split into 2 daughter cells, and you want each daughter cell to have a full set of organelles and DNA. So that's G1. The S phase is the DNA replication or DNA duplication phase. So you have to have enough DNA for 2 cells, because eventually you're going to split into 2 cells. G2 is another subphase and interphase. You're doing more of your cell job. You're preparing for mitosis to divide. And then the M phase here, you're dividing the cell into two equal diploid daughter cells. So diploid just means that each daughter cell will have the same number of chromosomes as the parent had. In other words, each daughter will have a complete set of DNA. And in this cartoon, we said it was 1, 2, 3, 4 chromosomes. This looks like there's 1, 2, 3, 4, 5 chromosomes, but there should be 4. We'll have to call the people who drew this cartoon. The M phase, or the mitotic phase, has 2 parts. Like we said, it's got mitosis, cellular division. You need to do it for growth and tissue repair. So for example, your oral cavity cells are always being abraded when you eat food, so you have to make more of those. You need to have a process of cell division in your oral cavity. And then the second phase of mitosis is cytokinesis, when the cytoplasm is actually divided. All right, so let's look at the phases in more detail. So DNA replication or duplication occurs in the S phase, or synthesis phase of the cell cycle. So here's DNA. So this ribbon, this purple ribbon, is a sugar phosphate backbone. And then here's the bases. A always pairs with T, and G always pairs with C. And these little dots here between G and C, for example, those represent hydrogen bonds. And we talked about those when we talked about chemistry, so hopefully you know what those are. And then, so it shows here, if you follow this, we've got an old strand or a template strand. So if we follow this back up, this is the original strand from the mother. Okay, and here's the other original strand from the mother. And then what you do is you build a new strand, and that's what it shows in this cartoon here. So see how it's cut here, cut here. This is the new strand of DNA being made, because probably know DNA is a double helix. So each daughter cell has to get a full one of these double helices. So in S phase, the DNA uncoils, and that's what this is showing, that at this fork right here, the DNA, the duplex of the double-stranded or duplex DNA has uncoiled into 2 nucleotide chains. So these are the nucleotides in the chain, A, T, C, and G. And each chain serves as a template for a new DNA, and we looked at that. We looked at the template strand. And we said this nucleotides are always complementary. A always pairs with T, G always pairs with C. And this bond here is specifically, it is a hydrogen bond. So for example, if this sequence was in the template strand, T, A, C, T, G, C, then the new strand that formed off the template strand would be what always pairs with T? A. What always pairs with A? T. What always pairs with C? G. What pairs with T? A. What pairs with G? C. What pairs with C? G. So this would be the template strand, and then this would be the newly synthesized strand of DNA. All right, so this shows a cartoon of interphase, and then remember there's subphases within interphase, G1, S, and G2. So here's a cell. It shows in G1 phase. It shows here chromatin. So chromatin is just DNA that is unwound or loosely wound. And then here's these guys. The centrioles will play a role in cell division. G2 looks pretty much the same way as G1. And then here's S phase. We just talked about that, where you're synthesizing a new strand of DNA. And we said this earlier, the sequence of phases in mitosis: prophase, metaphase, anaphase, and telophase. Okay, so let's look at these in turn. So we're here, we're in the M phase, and then a subphase within the M phase is prophase. So we're going to look at that, and there's actually an early prophase and a late prophase, so it gets even more complicated. But in prophase, so here's our hero, here's the cell, here's the mom cell, and remember this mom cell wants to divide into two daughter cells. So in early prophase, the chromosomes were already duplicated, aka replicated, in the S phase of the cell cycle. So check it out. You have there's a template strand of DNA, and then the newly synthesized strand of DNA. And now, instead of being in chromatin form, the DNA is in chromosome form, so it's tightly wound up. And the chromosome, actually, because it's been duplicated, the chromosome consists of two sister chromatids. So here's one sister chromatid, here's another sister chromatid. And the sister chromatids are held together in the chromosome right here by this thing called a centromere. So this gets a little tricky because there's a lot of C words. So there's chromosome. That's what these guys are. It's the DNA. There's a centromere. That starts with a C. That holds the sister chromatids together. And then there are centrioles. That starts with a C. And that's going to actually, the centrioles will direct this thing called a mitotic spindle. The mitotic spindle is here. The mitotic spindle will actually allow the cell to split into two daughter cells. Okay. Now we talked about this chromatin will coil into chromosomes in early prophase. Said chromosomes are held together by a centromere, and a chromosome has two strands on it, each called a chromatid, or you could call them sister chromatids. It's the same thing. Okay, this is a late prophase. So look, now you can see, well, we still got our centrioles that are going to direct the mitotic spindle that will split the two cells apart. Within the mitotic spindle, you have these things called microtubules, and we looked at those when we did some basic cell biology. So here's microtubules. So the microtubules are extending out from the centrioles, and the microtubules within the mitotic spindle are attaching here at these centromeres, and the centromeres are holding the sister chromatids together. That's a lot. I know. You have to get your Zen candle out, look at the cartoon, and think about this. Think about the ultimate goal is to split one cell into two cells. So you have to do all this cool molecular biology. So the centrioles, which are here in late prophase of mitosis, the centrioles migrate to the poles. So we could say that's the North Pole, we could say that's the South Pole. And then they're going to direct the assembly of these mitotic spindle fibers, which are made of, technically they're made of proteins called microtubules. Okay, so the spindle itself, the mitotic spindle, so the mitotic spindle includes the centrioles and all these microtubules radiating out from the centriole. The spindles provide scaffolding, so everybody probably knows what a scaffold is, but they're scaffold for the attachment and the movement of chromosomes during later stages of mitosis. So remember to keep the big picture in mind. You've got these duplicated chromosomes here, and each daughter cell eventually will get one set of each one of these chromosomes. So one daughter cell will get this sister chromatid, will get this sister chromatid. The same daughter cell will get this sister chromatid, and we'll get this sister chromatid. And then the other daughter cell will get this sister chromatid, this sister chromatid, this sister chromatid, and this sister chromatid. Also in late prophase, the nuclear envelope breaks down, and that's what these purple pieces are. This used to be the nuclear envelope that held the DNA inside, but it breaks down, and it has to break down because you got to form 2 new daughter cells. All right, next stage of mitosis is metaphase. In metaphase, the chromosomes, so that's these purple things, they're chromosomes, but they're in sister chromatid form because they're duplicated chromosomes. The chromosomes are aligned in the center of the cell. That's what these dashed lines show you. That's the center of the cell, and they're all lined up. And this plate, it's an imaginary plate. It's called the metaphase plate, and that's where all the chromosomes line up. But remember, the chromosomes, they're still duplicated. And when were they duplicated? In S phase of interphase of the cell cycle. If you said that, you're a genius. Okay, the metaphase plate, which is that imaginary plate, kind of like there's an imaginary equator on the Earth. There's this imaginary metaphase plate in the cell, and its center, it's right in between the centrioles. So here's the North Pole with a set of centrioles, South Pole, and then the equator, aka the metaphase plate, is midway between those 2 poles. And then it says here a straight line of chromosomes in sister chromatid form, like we talked about, is seen on the metaphase plate. So that's metaphase. And then the next phase or stage of mitosis is anaphase. And you can see that these microtubules, well, they're spindle fibers, but what they're made of is microtubules, are attached to these centromeres on the sister chromatids. And then these microtubules are linked up to the centrioles here. And what happens is you have a lot of movement, but basically each one of the sister chromatids is being pulled to one pole, because remember, you're going to get one daughter cell, will get all the chromosomes here, the centrioles, and everything else in the cytoplasm, and another daughter cell will get all this. Anaphase is complete when the chromosomes stop moving. Then after anaphase, next stage of mitosis is telophase or telophase, however you like to say it. It's kind of like the reverse of prophase. So the chromosomes, which are here, now they're single-stranded. They're not duplicated anymore. They're single-stranded. They start to uncoil, and an uncoiled chromosome is called chromatin. The mitotic spindle starts to break down, so all those microtubules that were coming out of here, those break down. And then you see this purple stuff, the nuclear envelope will start to reform around the chromosomes of each daughter cell. And then cytokinesis is division of the cytoplasm, and it completes during telophase, at the end of telophase, when you get 2 new daughter cells. Yep, and here's the whole thing. In most cases, mitosis and cytokinesis occur together. So they usually say telophase, which is the last stage in mitosis, and cytokinesis usually occur together. In some cases, the cytoplasm doesn't completely divide. This is commonly seen in liver cells. Kind of interesting. And mitosis gone wild. So uncontrolled mitosis is the basis for tumors and cancers. So it's really, mitosis, I know, mitosis is kind of interesting because you probably learned it in high school and you're like, okay, I'll just memorize this stuff. And then you're learning it again now and you're like, oh, I remember seeing this. But it's really important that scientists understand mitosis and understand how carefully it's regulated, because when it becomes dysregulated and the cells start growing and dividing out of control, you get tumors and cancer. And this here's a bigger picture of the cell cycle and mitosis. And that's the end.